So my name is René Paul. I'm affiliated with the Department of Biological Psychology here at Vrije University, and this department is hosting the Netherlands Twin Register. Here you see the logo of the study that holds data of twins and their families, so monozygotic twins and dizygotic twins. Yeah, and I think in total it's about 200,000 people that we have at least one data point of. Wherever you see NTR, so Netherlands Twin Register, in this presentation, yeah, you could also read your own study or just a study or a cohort where you want to perform polygenic scoring. So that's the topic of today, polygenic scoring. I'll explain what that is and how you can determine polygenic scores, and I'll finish up with an application. So on the bottom right, you see a link, and I'll click on it. It's an explanation of what polygenic scores are, but then for the general audience, and I think it's a very informative website. So I'll just go over it. As we know from our course, we all have our own set of DNA, all of our cells, and this the sequence of letters in our DNA is fixed. It's static. It's the same in every cell, except for some variations or mutations. And there are differences between individuals. So that's shown over here. So we have two persons, and here you see at a single base pair position, you see a varying point mutation or a point variation. That's also called a single nucleotide polymorphism or a SNP. So something that varies between individuals. I'll come to how you determine data of this picture over here. But what you see over here is that some people have a shared variation at a position, and some of them have only, for some variations, only one person has the variant. And what is signified over here is that these orange variants, these orange V's, are they influence the risk for a certain disease. So the genetic risk for a disease. Yeah, the way it influences disease can be either positive or negative. So it can be a risk increasing variant or a risk decreasing variant. We've seen in our course that there are some diseases that have only a cause, or how should I say it? They are caused by only single genes. So variations in a single gene, and one example is cystic fibrosis, where there are variations that cause mutations at the protein level of this gene, this transmembrane conductance regulator gene or protein. So this is not polygenic. So this is monogenic, so to say, single gene. But most diseases or most traits are complex, and with complex I mean they have, if you look at it from a genetic point of view, there are small contributions to variants across the entire genome, as you see in this picture. So this is a picture for artery disease, coronary artery disease, and you see that there are, across the whole genome, there are tiny or subtle effects that, if you add them up, might lead to really an increased risk, or maybe a decreased risk, depending on what type of variant an individual has. This is a distribution of polygenic scores in in the population. So most people, for a given disease or a given trait, most people will be somewhere here in the middle of this distribution, and some of them have a decreased risk at the left-hand side of the distribution, and some of them will have a high risk on the right-hand side of the distribution. And of course, if you know what the population average is, you can determine for an individual if they are on the right-hand side of the distribution or on the left-hand side. I'm now going into how you actually calculate a polygenic risk score for an individual or for an entire study like the Netherlands Twin Register. I took these pictures from this publication, Nature Protocols. It's a very nice paper that it's actually a tutorial on how to generate polygenic risk scores, and there's also really a tutorial site on GitHub that is accompanying this this paper. So if you're interested in polygenic risk scores, yeah, I advise you to check out this paper. This is the central figure from that paper, and you actually read it from top to bottom. So on the top you see that we have base data and we have target data. So target data in our case is the Netherlands Twin Register, but you could also fill out an individual over here or maybe a different study that you're working on. And the base data is a publication on a genome-wide association study of your trait of interest. This is a different representation of the base data. So this is based on genome-wide analysis of a certain trait, in this case, coronary artery disease. And such a association study highlights where there are risk increasing or risk decreasing variants. So these are the 22 chromosomes and maybe also the sex chromosomes, and the height of the dots. Every dot is a variant, and the height of these dots says something about the significance of the association. So I'll go through this figure step by step. As I just told you, so this can be the summary statistics of a genome-wide association study, the GWAS, for a trait of interest. And I'll also briefly explain what a GWAS is, so don't worry if it's unfamiliar to you. And over here, the target data for in my case is the subjects that are part of our study, the Netherlands Twin Register, the NTR, that have genotype data. It's very important that you already see it over here that these samples should be independent. So often genome-wide association studies are published as meta-analyses, and you should really make sure that the base data you have does not include the target data. Otherwise you get inflation of your downstream statistics. So that's very important. So the base data should not include the target data. The base data, so this is actually the GWAS, or genome-wide association study part, and this also touches upon what we discussed during our course, so some molecular genetic techniques. So you start out with data collections, sampling people. In this case, we're interested in DNA, so you could take saliva or blood, and using those samples, you can genotype these people by performing sequencing experiments, whole genome sequencing, or using array techniques, much cheaper and faster, to genotype people and get to know their single nucleotide polymorphisms, because that is also, those are the variants that we are talking about in genome-wide association studies, single nucleotide polymorphisms. So once you have your genotyping done in your entire sample, you need to quality control them, and then you can impute the data. You can actually predict variations of unmeasured SNPs by using information on the correlations within our genomes. So there are statistical packages and softwares to do this. So the genotyping, if you do that on a chip, you get, well, approximately 500,000 variants. By using imputation, you can explode that actually to, well, tens of millions of variants. And those you can use for association testing. And the association testing for one SNP looks like this. Each data point in this graph is one subject, and on the x-axis it's denoting their genotypes, and 0 signifies the AA allele combination, 1 the A small a allele combination, and 2 the small aa combination. And those, of course, reflect the parental genotypes that we inherit. And on the y-axis, we have our phenotype, and that could be the concentration of glucose in your blood, also measured in the same sample. And then you can do a linear regression. Here's the formula. So the phenotypic score is regressed on the SNP, the x-axis, and we get an estimate of the effect size beta. So that's what we're interested in. And if you do linear regression, you also get a p-value for this effect size. So sometimes it's not significant and sometimes it's highly significant. And that is actually what is plotted in this graph. This is called a Manhattan plot because it resembles the skyline of Manhattan. On the x-axis, we have the chromosomes, so the millions of SNPs for which we do linear regressions. And on the y-axis, we have the significance. That's the minus log of the p-value of the effect. And the higher the dots are, the more significant the association. So this is how you get to know where there are risk variants in our genomes for a certain trait. For the target data, we need a sample or a single individual for who we do genotyping, quality control, and imputation. And using the results of the target data, we now can do the scoring. Yeah, but before that, you have to do a quality control step. So you should take away variants that have a low imputation quality or have a very low allele frequency, stuff like that. Yeah, this is often a time-consuming task because you have to sometimes communicate with the authors of the people who generated the base data. So yeah, this can take time. Now remember that the variants in our genomes are correlated with each other. So that's exactly what we take advantage of with the imputation I just told you about. And the reason that associations are often not independent is shown in this slide. So it really comes from the meiosis cell division to generate germ cells. So what we know, the result of meiosis, that gives you haploid germ cells, and each of these germ cells has a different composition of the DNA, and that's the result of independent assortment. So the mixing of maternal and paternal chromosomes, And as we know from the course, recombination. So that's the exchange of paternal and maternal DNA between the sister chromatids of homologous chromosomes. This exchange goes in chunks, as shown over here. And that's why variants that lie together on this chunk over here get inherited together. So there is a higher correlation between variants lying on this chunk as compared to a variant on this chunk and a variant right over here on the other side of the chromosome. And the term for this correlation is called linkage disequilibrium, LD. So if a variant has a high correlation, if it really gets inherited together, the LD is high. So, and if it's low, the correlation is also low. Yeah, this is an example of a recombination pattern in sperm cells as a result of meiosis. Okay, let's go back to polygenic scoring. So we now know that SNPs are correlated, so their associations are also correlated, and we need to adjust for that. So we can do something that's called pruning. So only take into account the lead variants for a certain location on the genome. So that's on the right-hand side. It's also possible to do beta shrinkage. So that's actually a statistical method to reduce non-significant or non-contributing betas to 0. So the result is that you are left out with less variants, but the ones that you retain are significant and have a non-zero contribution. And there's also a method or methods that you can use to weight the effect sizes by their LD structure. And methods to do that are called LDPRET or LDPRET2. And sometimes you also use a method called S-base-R. Yeah, and then after you have taken out the independent signals, you can perform the actual polygenic scoring, and that's shown in this slide. So let's say here on the top we have 4 point variations, 4 SNPs, and we assume that they are independent. And then on this row, their effects are shown. So if you have an A allele, then the score is increased by 1.5. If you have 2 A alleles, it is increased by 3.0. And the same for other polymorphisms over here. And an example is shown here for 4 individuals. So we have individual 1, 2, 3, and 4, and these are their genotypes for those positions. And then if you do the scoring for those positions, so individual 1 has 1A, so that's plus 1.5. It has 1C, so that's minus 0.5. It has 2 T's, so that's 2 times 2.0, so that's 4. And it has 0 A's, so that's a contribution of 0. So the score of this individual for this trait is 5. And we can also do that for all other individuals in the sample. And then we can plot a histogram or a distribution, and then we see that the individuals in this example are located at these positions with respect to the population. Yeah, and then you can do all kinds of extra steps to validate the score with respect to the general population. There are several methods to do that. So if you're interested in doing PRSs, or if you have to do them because you need to have them for your analyses, the top two steps are hard work for you, because you cannot really automate these steps. And the bottom ones are hard work for the computer, so you need to have a lot of compute power. So at the Netherlands Twin Register, we have a Polygenic Risk Score archive that's maintained by me, and it's also a growing archive, because I often get requests to calculate a PRS. So for many traits, we already have these scores for our study. Yeah, I think it's now 260 traits or something like that. Yeah, and this is an example of some traits for which we have polygenic risk score. So you see, for instance, over here, 2 times ADHD. That's the trait. And here the target, or the base data, was published in 2 papers, one in 2019 and one in 2016. So often people would like to have the most recent one, so they choose this one, and then I generate the data set. Yeah, maybe that's a question to you. Why would it be handy to generate a polygenic risk score if you are doing population-based statistical research on your study? Well, let's say you want to study BMI in your cohort, and how that is, for instance, affected by smoking behavior, or age, or whatever. And what if you don't have BMI data, but you do have genotype data of your participants?
Like basically to calculate the risk of a certain disease.
Yes, exactly. Yeah. Yeah, that is that's a very good answer. Another way would be, so I took the example of BMI. It's a phenotype that is easily measurable. But if you don't have it, you can do, and you do have genotype data, you can generate polygenic scores, and then you have a proxy phenotype. So that's actually also how you can use it in population-based research. And again, BMI is very easy and cheap to measure, but maybe MRI measures of brain volumes is very hard and expensive to measure. But there are GWASs on brain volumes, so you can generate a proxy phenotype for your research question. But if you have the need for a polygenic score for a certain trait, they have to be calculated. So the recipe is that you have you provide quality, or generate quality controlled GWAS summary statistics. So those are the base data, excluding your own study, in this case NTR. If NTR was part of the discovery, then you should contact the authors of the paper and ask if they can do a meta-analysis without the study, without your own study, without the study for which you want to calculate the score, so the target data. Yeah, you need to know some more details, the original paper. And this is actually, the last point over here is what keeps me off the street, because every study published in a journal, yeah, likes to use their own column names for some reason. So this is not standardized or harmonized yet. So I have to, if I see so many statistics, I have to deduce what is the chromosome column, what's the position column, and so on and so on. And sometimes there's missing data, or people write the allele names in small caps instead of uppercase letters, and so on and so on. Then some words on compute resources, and you have to think also about this when performing these scores. So you can you have to take into account the sensitivity of the genotype data. So you cannot just have the genotype on a laptop. You also take with you on the bus or the train. So this is highly sensitive data because it's very identifiable, right? If you know the DNA of a person, you can almost identify people on postcode or street level. So it's that sensitive. So we at our department have our own resources, secured compute servers over here. And there are also often, for instance, the Amazon services or Microsoft Azure services that you can use to get compute power. And in the Netherlands, we have our own supercomputer called Stellius, where you can also perform calculations. But on those compute environments, you cannot really store the very sensitive genotype data. So you can handle, for instance, the base data on such machines. But if you are going to perform actual scoring, for which you need the genotype data, you have to do that in a secure environment. Stellius is the national compute cluster, also located in Amsterdam. But for our study, we don't have access to the genotype data to perform the last step, the actual scoring. So that's why we perform those calculations on our own resources. And it also hosts the genotype data. Yeah, for handling all of these data types, you need to have high memory and a lot of compute power, because yeah, what you have to handle millions of variants and thousands of samples. So that's why you need a high memory machine and you need a lot of compute power. Okay, so this brings me now to the last part. So an application of PRS or polygenic scoring. But before that, I would like to ask you if there are any questions up to now.
I was wondering if there's a difference between the NTR Compute 1 and the Compute 2.
Oh yeah, no, they're, for the user, they are quite similar, but the hardware is a bit different. So for the first one, it's Intel chips, and the other one is AMD chips, stuff like that. But they're not very different for the user.
Right. Thank you.
Yeah, what it actually looks like is, if you log on to the machine, it looks like this. It's a terminal. So this is what it looks like for the user. Yeah, I want to now go into an example of an exchange I did a few months ago within the exact project, the European project, that was aimed to have staff exchange or to enable staff exchange in the healthcare system around Europe. And the idea was that people from academia could have an exchange in industry and vice versa. And for that project, Carla van El is somewhere over here on the picture, and Veronica made it possible for Veronica and me to do a secondment, an exchange in the UK. So we went to Alelica and I stayed there. Alelica is a company, and Veronica went on to the PhD foundation in Cambridge. Yeah, so these companies and institutions, Alelica and PSG and the VUMC are partners in the exact project. So me and Veronica were seconded at Alelica, where we compared performance of different polygenic scoring methods. And we also were aiming for looking for ways to communicate the results of PRSs to the general public, maybe patients or medical doctors. And Veronica went on to Cambridge, and during her secondment, she interviewed experts in the field of generation of PRSs, so geneticists, but also more in the field of applications there. So maybe clinicians. Yeah, so the products are not yet finished. We have to generate reports, and maybe we can generate a nice paper. So now I will now focus on the secondment at Alelica. So this is a PRS company, actually. So this is a company that wants to bring polygenic scoring to the clinic. Yeah, that requires some extra work, or a lot of extra work, so to say, as compared to the picture I showed you of how to generate the PRSs. You now also have to really validate polygenic scores, and you should prove or show that it has added value to the clinical practice. So that's where this company is aiming for. And the secondment I did was a technical one, actually, comparing PRS methods and try to find out ways of how to communicate those. As a trait of interest for disease, we took major depressive disorder, which is a disease where you have to, so the phenotype is determined by this top row. So you have to have at least a 2-week period of low mood, so low self-esteem, loss of interest or pleasure in activities that you usually find enjoyable. So it is treatable, for instance, by psychotherapy or by medication, but success rates are highly variable. The disease burden is high for the affected people, of course, their environments and society. So there's also a high cost involved because people, if they are severely affected, cannot work anymore, for instance. looking at it from a genetic perspective, it's a complex trait, a complex phenotype. So a lot of variants are involved in the disorder. Genetically, the heritability is moderate. So I think it's up to 40% in the general population. So that means that the phenotypic variation that you observe in the population is explained for 40% by variation in the DNA, in the genomes. And it's a highly polygenic trait. And that is shown in this slide. On the right-hand side, you see the GWAS results for lifetime major depressive disorder. And on the x-axis, again, you see the chromosomes, and each dot is a variant. And the height of the dot signifies the significance of the association. So the higher the dots, the more significant. This picture is from a previous meta-analysis, and the total sample size was about 800,000 people. There's a new initiative where there are 3 million people involved, so 3 million subjects divided over cases and controls, based upon which we can identify even more locations on the genome that contribute to the disease. We already took the base data from this last effort to do our work. And this actually is the schematic of what we did and what we're still working on actually. So on the left-hand side, the aim was to build PRSs using different methods. So that's over here, number of methods. And for that, we need the base data, GWAS, and the individual level data. That is the genotypes of the individuals in NTR, and for Allelica, the genotypes in the UK Biobank. And to be able to assess the performance, we needed to have the phenotype itself, and we also have that for the UK Biobank people and for the Netherlands Twin Register people. We generated weighted betas from which we calculated PRSs, and then we tried to validate these PRSs by several performance characteristics over here. So maybe explained variance, area under the curve in received operating characteristic analyses, and maybe odds ratio. We're still working on that. For now, we took the R-squared or the explained variance. So that is when you have the final PRS determined by some method, one of the N methods over here, and you calculate how much does this PRS explain the actual phenotype, so the case control data that we have for NTR, and you can express that in a R-squared statistic. Then the idea was to choose the best PRS method, or maybe do a meta-analysis of several methods, and then have a test sample in NTR and another study that we closely participate with, NESTA, the Netherlands Study for Depression and Anxiety. And we also tested, or are aiming to test, in UK Biobank. In the end, we obtained a Dutch and a UK PRS, and yeah, we can actually also include differences between these two countries with respect to the PRSs. So this is some preliminary results, a table with a lot of numbers. So we have to focus on this column, so the left one. These are several methods, LBPRED1, SCT, S-base-R, and PRS-CSX. So those are PRS generating methods. And then here we have the variance explained. So I think we should take the most right column. So the variance explained is something that varies, and there is a maximum for LDPET 1 over here. So this is 3.7% of the variance in the phenotype is explained by the PRS. This is something that's not unusual. It's something you would expect. But you see that other methods, like over here, PRS CSX or SCT performs much better. So using these kinds of statistics, you can actually determine what method works best, and maybe in the end we will take a combination. The next step is how to present those scores and translate them to individuals or read patients in the clinic. So for me, the PRSs are a research tool for which I do population-based research. But of course, in the clinic, you want to help out a single patient. And for that, you actually need to know the absolute risk a person has with respect to the population. And then we come to figures like this, so distributions of PRSs, and then you want to locate the client or the patient that you have in front of you, if they have a low genetic risk or a high one. Maybe you can even assess the risk increase as a function of age, shown on the right-hand side over here. So these are things that we still have to tackle and to find out for our particular phenotype. I would like to finish with maybe we can have a short discussion on the question over here on the bottom. So when would a PRS for depression be useful in the clinic? So again, for me, it's a research tool, but how applicable are they in the clinic? So what are the conditions needed to have these PRSs as a useful tool? So I hope you have some ideas, and we can have a short discussion and finish up.
Well, wouldn't it give an idea of like how likely you are to have a disease in some way?
Yeah.
So like you could maybe prevent it if possible.
Yeah.
Yeah.
Yeah, exactly. And then the
Okay.
question is, can you, so this is determined from your DNA, and DNA is static.
Mhm.
So it would give a lifetime risk, so to say.
Yeah.
Would it help if you know that you are at high risk for depression?
I feel like in some cases, yes, it might depend on the disease. For like depression, because you can take preventable action against like making the depression worse, let's say.
Yeah.
Or like if you're more at risk for heart disease or like lung disease, then I wouldn't smoke, you know?
Exactly. Yeah. Yeah, I would agree. And stuff you also have to take into account is the heritability of the trait. So this is moderately heritable.
Mhm.
So maybe the risk doesn't really, the genetic risk doesn't really say something, because, you know, 60% is explained by environmental factors. But for instance, schizophrenia is very heritable. So a score for that trait would make it more actionable.
But if there were no treatment method, then like PRS can also help like study new ways to find treatment or prevention methods. No?
Yes, I agree. Yeah.
Okay.
But then you would be, that would be more research and not really presenting it to the patient, I would say. Yeah, so actually that is the reason why these PRSs are used for diseases where you can take action, for instance, heart disease or cardiovascular diseases or type 2 diabetes, because those are really preventable, right? You can change your lifestyles, for instance, if you know you're at high risk. So in the field of psychiatry, I think the acceptance of genetic information is a bit lower as compared to other fields in a medical practice. But I'm not really sure. I don't know. I think that is the case, but I don't have any proof or evidence for that. Because indeed, this was my last slide.
I think maybe I missed the whole point of this, but are we comparing like the target data, so the NTR, to a GWAS study? Like, how does that work? Like, how do you not have the NTR in the base data?
So again, the question is, why should you take out the NTR? Or,
Yeah.
or was it a different question? Maybe I misunderstood.
No, that's my question.
Yeah,
Because you said the NTR like should not be included in the target data.
yeah.
Which that makes sense, but how do you take it out?
Yes.
Wouldn't it be
Because-
part of the data?
So there are methods to, if you know what you have submitted to the meta-analysis team, then there are methods to subtract that that file that you submitted from the meta-analysis. That's kind of a reverse engineering of a meta-analysis. And the other way would be to ask the meta-analysis team to repeat a meta-analysis, but then skip the NTR file.
Okay. But then wouldn't they be missing like the genotype that you're looking for?
Yeah, so the results will become less significant, indeed. But since you're looking for polygenic signals, because it's in the name, and you will catch those if the remaining sample size is high enough, then you will catch those signals also in the resulting PRS. The risk of keeping NTR in while scoring in NTR is that all p-values of subsequent calculations will be highly inflated. So
Right.
you will find much more significant results, false significant results, if you would have kept NTR in. So that's really a danger. So it's always better to be on a safe side in that respect.